Hello everyone, warm welcome to this session, wonderful session. In this session we have to discuss Chapter 10 Part 3, and this Part 3 will include significance of mitosis. Then we have to see detail of meiosis. You know, meiosis is having two parts, meiosis first and second. In this part we will discuss detail of meiosis first. This is part that is 3 of Chapter 10. First we have to see in this part significance of mitosis. You know, mitosis means what? Number of the chromosome in daughter cells and genetic constitution of the daughter cell is equal to the parental cell. Therefore, it is called equational division, and it is usually restricted to diploid cell. Very important one, usually restricted to diploid cell. However, in case of lower plants and some social insects which have only haploid number of chromosome, that is, they also divide by mitosis. The question, you got it now? That is, mitosis is usually found in diploid cells. Number of the chromosome in diploid chromosome will be 2n. In daughter cell also that will be 2n. That is in case of that is lower plants, and in case of some social insects it is found in that is haploid cell also. It results in production of diploid daughter cells which are identical in genetic constitution. Number of the chromosomes are equal, as well as that is their genetic constitution is similar to parental cell. Growth of multicellular organism is due to mitosis. I told you, we can take our own case also. We started our life from single cell, that is zygote, having 2 and number of chromosome, that was denoted as zygote. Mitosis division from 1 to 2 cell, 2 to 4, 4 to 8, 8 to 16 and so on. And then trillions of the cells have been formed, that is multicellular organism have been produced. Cell growth result in that is disturbing the ratio. I told you nucleus and cytoplasmic ratio, nucleocytoplasmic ratio is to be maintained only by that is mitosis cell division. It restores nucleocytoplasmic ratio. If there will be no mitosis, then a time will come every time, you know, nucleus will divide in two, and in the last nothing will be left. That's why mitosis, it restores, yes, nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair, right? For example, in case of skin, you know, that is upper layer of the skin are being damaged, and that's why these are to be repaired by mitosis cell division of the cells which are found in that is layers beneath this upper one. Lining of the gut, blood cells are constantly being repaired. Mitotic division in case of that is, you know, in case of plants, that is mitosis division in meristematic tissue. Like this is apical or that is lateral, that is meristem is there, that is primary meristem or secondary meristem, apical meristem or lateral meristem. It results into continuous growth of that is plant, right? Now we'll proceed to the next important cell division, that is meiosis cell division. Meiosis is also known as that is reductional division. It is also called reductional division. Why reductional? Because number of the chromosome will be reduced to half. You know, if this is parental cell having 2 and a number of chromosome, it will divide into 2. Each daughter cell will have half number of the chromosome n, and again each will divide so that the 4 cells will be formed, each is having that is haploid number of chromosome. We have to see it now. The term meiosis was proposed by that is Farmer and Murray. Can you tell me where you expect meiosis cell division? It is found only in sexually reproducing organism. Special cells in multicellular organism which switch from mitosis to meiosis at a specific time in their life cycle. Then the important question here is, can you summarize where we don't have meiosis cell division? See 5 kingdom classification, that this was the Monera, that was Protista, that is fungi, that was kingdom Plantae, and that was kingdom that is Animalia. The question is, out of these, how many kingdom doesn't have meiosis? This is only one, that is Monera. In case of Monera, that is, there is no meiosis in Monera, remember, because they do not have sexual reproduction. So meiosis is found in sexually reproducing organism. Remember, my dear children, very important point. So meiosis ensure production of haploid phase. It means meiosis is always found in a diploid cell. This is diploid cell. Meiosis will take place only in diploid cell, as a result of which in first phase, that is, two haploid cell will be formed, N and N, and then there will be mitosis like division, so that each haploid will divide into, that is, four, that is, two cells. Ultimately 4 haploid cells will be formed having haploid number of chromosome. So on the basis of this organism where fertilization that restores, again these haploid cell will unite to make zygote and diploid phase will be restored. So in the beginning, can we tell, that is, some important key points, key features of meiosis. Number 1, you can see here 2 sequential cycle of nuclear and cell division. It is known as meiosis first. This is known as meiosis second. So how many divisions of nucleus we have? 2. 2 sequence cycle of nuclear division, right? But very important, single cycle of DNA replication. Here after first division there will be no DNA replication. Replication will be only after, that is, second mitosis or meiosis division. Okay? Meiosis first is initiated after the parental chromosome have replicated to produce identical sister chromatid at stage S phase. Very important, because the single chromatid cell, the chromosome, if it is having single chromatid, that won't enter into meiosis. Meiosis involves pairing of homologous chromosome. We have to see meaning of homologous chromosome. This is one chromosome has come from father and almost the other chromosome of the same structure, same morphology, it has come from mother. We can represent this is the, that is, chromosome having almost similar structure. So it has come from father, it has come from mother. So this pair is known as homologous pairing of chromosome. And after that there will be recombination between non-sister chromatid. This and this will be known as non-sister chromatid. And 4 haploid cell will be formed at the end of meiosis. Right? We have to say, that is, detail of this now. How can we summarize? Can you tell me now? Meiosis first, there will be karyokinesis, and this karyokinesis will have prophase first, followed by metaphase first, anaphase first, then telophase first. There may be, after telophase first, what will be? 2 nuclei will be formed. Each is having, that is, haploid number of chromosome. There may be or may not be division of cytoplasm. It is known as interkinesis. It is called interkinesis. Is it right? And after this, the cell has to enter into meiosis now. Meiosis second, meiosis, that is, metaphase first, anaphase first, and telophase first. Now the cell has divided into, that is, 4, that is, cells 1, 2, 3, 4. We have to see detail of this. What is important here? This prophase 1 is very, very important one, and the most dramatic, most important phase in which there will be recombination. So see, children, this prophase we can divide into substages. We have to see detail of this, but first you see the line sketch of this. These are denoted as, that is, leptotin, that is, zygotin, that one is pecitine, that one is, that is, diplotin, and this one is, that is, dikinesis. In these stages you see, that is, in zygotin there will be homologous pairing of the chromosome. In pecitine there will be exchange of, that is, genetic material in between the homologous chromosome, that is known as crossing over. And by the end of dikinesis, what will be there? That is, as usual, as the prophase is over, the nuclear membrane will disappear. The centriole that has reached to the opposite pole, nucleolus have disappeared, and now the chromosomes that they have, their 2 chromatids, these are the 2 chromatids, but the structure of these chromosomes will be different from the chromosome which was in the beginning. Can you find out the sequence? That is meiosis first, prophase first, leptotin followed by zygotin followed by pectine, then this is diplotin, and then this is dikinesis. What will come after? That is this dikinesis. After dikinesis, the cell has to enter into, that is metaphase first, anaphase first, telophase first. That is the proper sequence. Is it right? Any doubt? Such type of questions have come many more times. You have to follow the sequence. Now, we have to see meiosis first. Detail of this we have to see. Reductional division or heterotypic division this is. Why reduction? I told you that is in this one cell having 2n number of chromosome that will divide into 2 daughter cells will be formed, and each daughter cell is having n number of chromosome. There is reduction of chromosome by half. Reduction. Chromosomes is reduced to half. And meiosis first, as I told you, that is consists of 4 substages, 4 stages. Prophase first, followed by metaphase first, anaphase first, and telophase first. Prophase first, yes, prophase first of the first meiotic division typically longer, and this is more complex when it is compared to prophase of mitosis. As I told you earlier also that it has been further subdivided into following phases: leptotin, zygotin, pectine, diplotin, and then this is dikinesis. So what happens here? You see, children, that is in prophase first, we have taken, that is 4 chromosome, 1 chromosome and 1 chromosome, 1 chromosome and 1 chromosome. So here we have 2 chromosome let from father and 2 chromosome let from, that is, mother. So how many total chromosome we have? In this diagram, just to see, just to understand the complexity, we have only 4 chromosomes. The 2 are maternal and 2 are paternal. What is then in leptotin? You see, before onset of prophase, that is here, this is the nucleus, and here is, this is the nuclear, that is chromatin material. 2 type of chromatin material will be there in, that is, nucleus. One will be from father, other will be from mother. Now this chromatin material, that will divide into definite number of chromosomes, segments, thread-like structure, and each species is having definite number of chromosomes, so it will divide into definite number of segments. What is there in leptotin now? That is, during leptotin stage, chromosomes become gradually visible under light microscope. That is, it will divide into, for example, we have taken 2 chromosomes from this material, 2 from this. We are having total 4 number of chromosome, and they are making pair. This chromosome, larger one, and this very larger one, that will make one pair, and this smaller and this smaller will make other pair. So during leptotin in animal cell, end of the chromosomes are attached to one side of the nuclear membrane. That this is the nuclear membrane. You know, nuclear membrane is intact even at present. It has started disintegration. So these chromosomes are, one of the end is attached to the nuclear membrane, opposite to which is that lies centriole, which looks like a bouquet of flower. So that this leptotin stage, also known as, that is bouquet stage, or also known as, that is Darlington stage, discovered by Darlington. And after this, it is being followed by, that is zygotin phase. So we have taken, for example, only 4 chromosomes. 2 are from, that is, one parent, and the 2 are from other parent, leptotin. Now zygotin. Chromosome starts pairing. Chromosomes have become much more thicker now because of condensation, and they start to make pair. It is making of pair is also known as synapsis. Such paired chromosomes are known as homologous chromosome. You see, homologous chromosome, that this is one chromosome like this having 2 chromatid, and this is another, that is chromosome of the almost similar length, similar type. This is from, that is one parent, this is from other parent. They have become more condensed, their centromere is visible, and the two chromatids are there. It is known as homologous. Let this is the other pair of the chromosome, that this is smaller one, that is this is one, that this is the, that is other one. How to differentiate? That this chromosome is from, that is mother, and this is also from, that is mother. It is known as homologous pairing of chromosome. Is it? Important point here, because these chromosome in type 2 come, exchange of material will be there in between these non-sister chromatids. These are called sister chromatid. This one and this one will be known as, that is non-sister chromatid. So these non-sister chromatid, they will exchange their genetic material. We have to study. But to make this process easier, what is there? That in between these homologous chromosome, see here, electron micrographs, they have revealed that synapsis is accompanied by formation of a complex, and this complex is called 𝕆𝕆𝔼𝕆 complex. This 𝕆𝕆𝔼𝕆 complex will develop in between these two, so that it will facilitate that they should remain together by the time that the recombination is over. One more important point we have here, number of bivalent is equal to number of haploid chromosome. In case of, for example, human, we have 46 chromosome, they will make 23 pairs. These 23 pairs will be known as, that is bivalent. So how much number of haploid chromosome we have? That is number of bivalent is equal to, that is haploid number of, in human we have 23 bivalent, and that is haploid number of the chromosome is also, that is 23. These first two stages, that is leptotin and zygotin, these are relatively, that is short as compared to the 𝕆𝕍𝑒tin. 𝕆𝕍𝑒tin, this is the third stage now. Very important one. This 𝕆𝕍𝑒tin is characterized by recombination. See here now, that is, we can see here, that is in next page we have to say, that is, this is the nuclear membrane. It will start disintegration, that is centriole. It is reaching to the opposite pole, as I told you. Nucleolus, that also start disappearing in nucleus, and the cytoplasmic organelles, they also started, that is disintegration, disappearance. But the more important point here we have, that is, these are the two, what are these? This is, that is, these are the two chromosome, two homologous chromosome. So what happened in two homologous chromosome? These are being, that is, placed together by 𝕆𝕆𝔼𝕆 complex. That is, here tetrad is there, this is tetrad, 1, 2, 3, 4, that is, each chromosome is having distinct what? Chromatid. The stage is characterized by recombination, really the site where that recombination will take place between non-sister chromatids of homologous chromosome. I'll tell you the detail of this. What is there now? This is, see once more, this is the one, that is, this one is the, that is one chromosome having distinct chromosome. Let this is the other, that is chromosome, it is also having distinct chromosome, and these are the homologous pair, that is, these are the things. Here we have 𝕆𝕆𝔼𝕆 complex. Now what will be there now? That is, here that will be exchange of, that is, material like this. That is, this part of this will go to this, and this part of this will go to this. These are the two non-sister chromatid, and after this crossing over, result will be that this is one chromosome will be like this, like that this is the other chromosome. You can see diagrammatically, that is here, that is, this part of this has gone to this, and this part of this has gone to this here. Now that is, this is the part which has been exchanged, right? So enzyme involved in recombination is recombinase. Crossing over leads to recombination of genetic material on the two chromosomes. Recombination between the two homologous chromosomes is completed by end of 𝕆𝕍𝑒tin and leaving the chromosome linked at, that is, crossing over. These are being even now, that is, it has been exchanged, but these are found that is linked like this. What is 𝕆𝕆? Beginning of the 𝕆𝕆 recognized by dissolution complex. That is this synaptoid complex. The purpose is over now. That will get dissolved. Dissolution of synaptoid complex, so that tendency of recombined homologous chromosome of bivalent to separate. They will like to go, that is separate here. X-shaped structure. This X-shaped structure known as chiasmata, that is visible in oocyte of some vertebrates. Important that 𝕆𝕆 can last for months or even for years. The cell will remain in this stage. What is there in dikinesis then? This is the last phase. You can see here in dikinesis, this nuclear membrane that will completely disappear. As I told you, there is, by the end of dikinesis, nucleolus disappear, nuclear membrane disappear, and that is cytoplasmic organelle will also disappear. And this is dikinesis is transition phase. It will get converted into, that is metaphase first, right? This is about the prophase first, one of the most important phase, prophase first. Now you have to see, that is metaphase first now. Yes, metaphase first, the bivalent chromosome, they are aligned on equatorial plate. Yes, bivalent, this is one, that is chromosome 2 chromatid. This is the other, that is chromosome 2 chromatid. That is 2 metaphasic plate will be there. And in anaphase, now you see telophase, the same thing we have here, that is opposite to prophase. Nuclear membrane, nucleolus will appear, and it will be followed by cytokinesis, is it? And that is chromosome, they do undergo some dispersion. They may or may not be. Extremely extended stage is never achieved. This stage between the two meiotic division is called interkinesis. Generally this is short-lived, and there is no replication of DNA. Important one here is, and it is called interkinesis stage. Astral rays, spindle fibers disintegrate and disappear, and at the end of telophase it is marked, that is karyokinesis is over. What is there? Cytokinesis now, that is after telophase. Cytokinesis produce 2 haploid daughter cells. It may or may not be there. RNA protein synthesis will occur. In animal cell centriole, that is duplication will take place, but there is no DNA replication, remember this. Now you see diagrammatically how can we represent. Let this is the prophase first. What will be there in metaphase first? The 2 chromosomes, they have been arranged on 2 metaphasic plate. In anaphase, complete chromosome is moving to the opposite pole. So we have taken, for example, 4 here. So what will be there now? In anaphase first, that is 2 will go to this side, 2 will go to this side, is it? And then after telophase, what will be there? That is after telophase first, that it has to enter therein. It has to enter in, that is meiosis, that is second, which is almost similar to mitosis division. So this is end of part 3, chapter 10.